Awesome. Thanks, Katie.
Mhm.
Hi, everyone. My name is Divya Sripathi, and I've had the pleasure of mentoring Nurzahan C this past summer. When we first met, she showed me some in-depth and thorough research that she had already done on the topic of converting food waste to energy, and she expressed interest in chemistry. So we came up with a project idea that combined the two, through which she can do a hands-on science experiment that explores a complex series of chemical reactions in real life using the scientific method. Throughout the summer, I've been so impressed by Nurzahan's dedication to her project, as well as her enthusiasm and inquisitiveness. She always asked questions, and we would spend some time in our meetings just marveling together at how truly amazing the science was. That sense of wonder combined with her determination will take her very far to solve any scientific problem she chooses. But for now, I'm excited for her to share more about her project with you.
Mm, Thank you, Divya. Hello, everybody. My name is Nurzahan C, and I'm a rising 11th grader at Midwood High School in Brooklyn, New York. Next slide, please. So before we begin, let's talk a bit about my mentor, Divya Sripathi. Divya is currently located in Northern California, where she works as an analytics program manager at a company called Intel, which is a technology company, and it's really cool. But aside from work, Divya enjoys painting and reading, which is something we both happen to have in common, and it is also both of our first times working with magic. Next slide, please. So the project we both decided to do was a hands-on experiment, and we named it Hot Dogs and Methane, which we will I'll explain why soon. But we focused it around a research question we created called how does the anaerobic digestion of food waste affect the amount of greenhouse gases released into the environment? And for anyone confused, anaerobic digestion basically is a process by microbes in which when they consume food to create energy for themselves, but they also release greenhouse gases as a byproduct. And so the reason why I wanted to do this project in the first place is because I have done research on this in the past, and I thought it was really interesting to learn how food waste can affect the environment when they end up in landfills. And I've also created a website on that in the past where it is linked there. And this time I wanted to learn about it from a physical perspective. So we did this by doing our experiment inspired by a lab from tech engineering. Next slide, please. So this physical perspective included learning the chemistry behind anaerobic digestion. And for the sake of this meeting, I will just be sharing a quick summary of what happens. So in anaerobic digestion, there are basically four chemical stages. The first one is hydrolysis, and this stage uses water to break down complex sugar chains like starch, and it just turns them into individual sugar molecules, which are called monomers, and which are like glucose or fatty acids. So then these monomers then undergo the next stage, acidogenesis, which is formed by bacteria, and it's the fastest step in anaerobic digestion. So the bacteria takes the monomers and turns them into long chain fatty acids. And then these long chain fatty acids then undergo acetogenesis, which is an extremely important stage and is performed by another group of bacteria called acetogens. So what these acetogens do is they convert these long chain fatty acids into acetate, and they also make carbon dioxide and hydrogen. And then this is all done through fermentation, and which is just another chemical process. Then the final step is methanogenesis, in which methane gas is produced from the acetate or hydrogen and carbon dioxide by methane producing microbes. And if you look in that picture, you can see that the food waste in the landfill is kind of releasing methane and other gases, which is basically what happens to this chemical process. Next stage, please. Next. Thank you. So now that we've gone through the chemistry behind it, let's talk about what my experiment actually was. So in my experiment, I kind of wanted to see how if placing food waste into an environment would increase the amount of greenhouse gases made. So in order to do this, I kind of did need to have a whole ecosystem and environment. So I went to a pond and gathered pond sludge, which you can see in that picture. But what pond sludge is just the dirt that touches the water. And inside this dirt and this sludge is a lot of bacteria and microbes, which is the ecosystem that I needed. And then I placed them into three separate soda bottles, as you can see in that picture. So that's where all my microbes are. Next slide, please. So this is what the finished setup looked like. I will just be explaining what you were seeing. So basically, we have the three separate bottles with the microbes inside. I put 500 milliliters of the sludge into each one, and they're all labeled, as you can see. The one with the blue cap labeled diced hot dog has a finely chopped wiener hot dog inside to act as my diced food waste. The one with the red cap that is labeled hot dog has a full wiener hot dog inside to just act as my whole food waste. And then the blank is my control group. So this one has no food waste at all. And so the microbes are going to be eating the food waste. And then as you can see, it is connected through a tube and I sealed it off to prevent other like outside air from getting in. And when it is connected through this tube, it is connected to a water bottle that is in surrounding water. And these water bottles, I'm not sure if you can see in the picture, but there are measurements on it. So each one is 500 milliliters, just so I could keep up with measurements as I take the data. And so as the microbes eat the food waste or don't eat the food waste in my blank and my control, they're going to be going through the whole anaerobic digestion process through the 4 stages I just talked about. And they're going to be releasing greenhouse gases. And then that's going to flow through the tube and it's going to fill the water bottle up. If you can go to the next slide, please. So as you can see in this picture, this is week 7 or my final week. So if you look closely, you can see that the bottles have risen. This is because the gases started to fill it up, taking up a bit more volume and just making it rise a bit. If you look at the one with the blue cover, which was my diced hot dog, it rose the most. And then the one with the red cover, which is the full hot dog, rose, it was the second most. And my blank barely rose at all. It only rose 50 milliliters, while the other ones went over 100. And so this was my final week, and it's very important to note that at times the microbes were creating too much gas, and it was just like filling up the bottle so much that I had to reset it by opening the water bottle and letting the gas release, and then it would just reset and it would fill up with gas again. You can go to the next slide, please. So these were my results of everything at the end. So the way I calculated my results was at the end of every week when I checked on my bottles and I just looked to see through my measurements how much milliliters was released. I wrote it down and then I averaged it out. And at the end of week 7, I just added everything up. And my blank or my control group with no hot dog, no food waste at all, really produced 275 milliliters of gas. My full hot dog produced 1,130 milliliters, and my diced hot dog produced the most, being at 2,010 milliliters, which this also maps with my original hypothesis, kind of supporting my theory that adding food waste into an environment increases the amount of greenhouse gases released, which looking at those results, we can definitely see that my diced hot dog released the most amount of gas. Next slide, please. So now that we've talked about my experience, let's go through my magical experience a bit. Sorry, my experiment. Now let's go through my magical experience. So some highlights of this whole project was definitely setting up my experiment because it was a lot of fun just hot gluing all the tubes down, putting in the water and kind of seeing it come to life. And another highlight was definitely seeing the results because I wasn't completely sure if my whole experiment would even work. So seeing the gases start to become created and matching what my hypothesis was a lot of fun and it was just really cool. Another highlight was learning about the history of anaerobic digestion because my mentor Divya and I were very fascinated by how people even found out about all of this in the first place. So learning about that was really cool. Some lessons was the scientific method, since that can help me in future instances where you just for scientific experiments, and the chemistry behind anaerobic digestion, which I walked you guys through. That was just really cool to learn about it from all the formulas and the exact chemistry. And then some challenges that I did face was gathering materials because I did have to find a pond area where I could gather a bunch of dirt without having people just look at me the whole time. And another challenge that I faced was keeping up with the data because each bottle was releasing different amounts of gas at different times. So it was hard to keep track with it. And a final challenge is external factors because I did have to move my experiment to a different location, which led to different temperatures. And so that I was worried that it wouldn't work, but it ended up all being okay. Next slide, please. So thank you for listening. If there are any questions, feel free to ask. And thank you, Magic, for this opportunity to conduct my whole experiment.
Wonderful. Thank you so much, Nurzahan, for walking us through your experiment. and all that you learned from it. Are there any questions from the audience?
I have a comment and a question. I think, like all other presentations, this is phenomenal. Nurzahan and Divya, you've done a very interesting project. I think I might have missed it if you explained it, but can you explain to me why the cut hot dog releases more? Is it because the surface area is included?
Oh yes, sorry, I forgot to mention that. But let me just explain that real quick. So the reason why the chopped hot dog released the most amount of gas is because since I cut it up into very tiny pieces, there was a more exposed surface area, like Ira just said. And so since there was so much more surface area, it was much easier for the microbes to consume the food waste, which was making them do it at a faster rate and releasing more gas.
Thank you.
I just saw that someone left a question in the comments. So the challenges that I faced. So I would say challenges that I faced when developing the process was kind of just trying to make sure that I was doing all the steps correct and to make sure that the microbes were in an environment where they weren't like upset with what was going on around them because they are living organisms. So I just wanted to make sure everything would go properly. And the second question you said, what inspired you to create this project? I did a lot of research on this and created a research paper during last year in school, and I was just really inspired by seeing how much food waste there was in New York and how much people were throwing out. So I really wanted to know how that would affect an environment, which was a big inspiration in wanting to see for myself. Yes, Artie, thank you.
That was such an interesting project and experiment setup. I loved listening to you talk through it and explain it so succinctly. One question, if you were to repeat this experiment, are you curious to try different types of foods or a slightly different experiment setup?
Yeah, for sure. I think if I was to conduct it again, I would kind of try to get like bacteria or microbes from a different place because I'm not exactly sure what type of bacteria I collected since I didn't check. But I think it would be super cool if I got like bacteria from a completely different environment or using a different piece of food, like you said. I think it would be really cool to see if the results would be any different. So yeah, thank you for your question.
Great job.
Well, thank you so much, Nurzahan, for clarifying